Carlos Campos from the Cawthron Institute, who will talk about some sanitary profiling and fecal source tracking tools that he's been working on. Dr. Carlos Campos is a senior water quality scientist in the coastal and freshwater group at the Cawthron Institute, and he carries out research in the domain of health-related water microbiology, particularly on catchment sanitary profiling and environmental and human health effects of water discharges. Now, Carlos is also a member of the International Water Association, Water New Zealand, and the joint FAO-WHO expert group on the microbiology safety of water. So welcome, Carlos, and we look forward to your presentation. If you can share your screen, share your slides.
Thank you, Libby, and thanks everyone who's signed in today. And thanks also to the Center for the opportunity to present. So in this talk, I will explore the application of sanitary profiling and fecal source tracking tools to determine sources and transmission of pathogens in aquatic environments. Now, sanitary profiling and also FST are not new research areas. They are, although, becoming more important and can be adapted to a large number of applications in a food safety context. So a reminder of the ways in which microbial pathogens can enter the food chain. So this could be the production environment, this could be a farm or a fishery, and this is the focus of today's presentation. It can be the processing environment or the preparation environment, and this could be a food preparation service or facility. In this new era of smart food safety, our ambition is to leverage technology to create a safer and more traceable food safety system. So I will show you that sanitary profiling and FST can play an important role in meeting this objective. So the rates of foodborne disease in New Zealand have not changed much in recent years. What I have in this table is NZ and overseas estimates of a few selected illnesses due to microbial hazards. Campylobacter is a top priority food pathogen of concern, with nearly 7,000 notified cases in 2018. STEC and hepatitis A also continue to be the focus of our food safety program. But I should note that foodborne transmission is only one of the many ways by which humans can be exposed to these pathogens. Other ways include contaminated waters, animal contact, and person-to-person. For some pathogens, notification rates have increased because more labs have used culture-based or culture-independent diagnostic tests using PCR. So foodborne pathogens are very diverse, include many groups of viruses, bacteria, and protozoa. And it's, of course, not possible to test for every single type of pathogen that may be present in the environment, okay? On the other hand, the methods that we use to detect and quantify pathogens can be labor-intensive and expensive. Worldwide food safety systems are different in many ways, but one thing that they have in common is that they're based on compliance with fecal indicator bacterial standards. These indicator organisms have been used to indicate the presence of pathogens, but in reality, and we know that direct correlations between indicator bacteria and pathogens are rarely found. This is because indicators are excreted at constant rates, while pathogens are more episodic. So in the environment, indicators and pathogens have different characteristics in the way they resist to stress factors. So this next slide introduces the idea of sanitary profiling. Essentially, this is an appraisal tool, risk assessment tool, that gives us a human health risk assessment for primary production environment. Sanitary profiling really evolved from the HACCP principles, which were initially developed for food processing industries to ensure sanitary controls in production steps. So essentially, what we're trying to achieve is to transfer some of these controls, or most of these controls, from the factory back up in the production chain to the primary production environment. For products that are consumed raw or likely cooked, a sanitary profile could be the key element in keeping or ensuring food safety and consumer protection. Sanitary profiles now are commonly undertaken by local authorities in many places. When, for example, when classifying a body of water for shellfish production, then it can also be used to obtain baseline water quality data for recreational waters, for example, and for drinking water systems as well. So what does a typical sanitary profile contain? So essentially, sanitary surveillance and evaluation of the primary production environment and its associated catchment area. It could also include routine sampling and testing of waters and food for bacteria, viruses, or even chemical contaminants, and also classification of the primary production environment based on analysis of monitoring results. So when we talk about sources of contamination, what we mean by this, there are many different types of sources affecting aquatic environments. The sources of greatest impact really differ from area to area, and their relative sort of contributions in relation to different areas are also affected by meteorological effects, such as rainfall, for instance. Other key factors that we use to assign risk are the wastewater treatment processes that may be operating locally, and the presence of intermittent discharges, such as pump station overflows or combined sewer overflows. Tourism is an important factor because it affects the loadings of wastewater from wastewater treatment plants. And during certain times of the year, waste discharges from boats may also affect contamination levels. So now looking at the sanitary profiling for shellfish growing areas, this usually comprises a desk-based initial evaluation of all the information that's available on potential sources of contamination. This can be sources of human and animal origins. We may also undertake a shoreline survey to kind of verify the findings of the desk study. This could also include an assessment of the circulation of contaminants as affected by tides, bathymetry, or currents in the area. And in some cases, we may want to undertake a bacteriological survey or a short-term sampling program of water or shellfish to support the sanitary survey assessment before routine monitoring takes place. Sanitary profile really looks at the transport of contamination at the catchment level, not just the immediate food production environment. The objective really is to determine concentrations of indicated bacteria or pathogens in treated and untreated effluents, in animal waste from things like farms, and also from birds and other wildlife. The key factor here, as I mentioned, is rainfall, because it kind of provides the energy that's needed for contaminants to reach the coastal zone. And then once in the coastal zone, things like tides and currents affect transport to a shellfish growing area, for example, or recreational water. Other factors that affect abundance and distribution of pathogens are turbidity or light intensity, or important stress factors. So the most basic information that we need to kind of support the sanitary profile is microbial concentrations from potential catchment sources. Unfortunately, we don't have much peer-reviewed data on common fecal indicator bacteria for a range of effluents typical of New Zealand wastewater treatment plants, or even for septic tanks discharges. So we tend to use information from the literature. Also, typical concentrations from diffuse pollution sources are often not available from many areas. And perhaps the least understood types of wastewater discharges are the intermittent ones. Few of these actually have flow monitors, and most of them are regulated through an allowed spill frequency, and that provide potentially is potentially a challenge for us in assessing their effects. While we can identify pollution sources through the sanitary profiling, this tool really cannot identify the host source of microbial contaminants with certainty. For this, we will need to combine sanitary profiling with other source attribution techniques, such as fecal source tracking. The use of sanitary profiling and FST is now the basis of health risk-based approaches around the world. And some of the most common applications of FST are, for example, identification of the main sources that may be contributing to water quality deterioration, or to determine which sources are the dominant ones in the catchment of interest, or really to focus management action through more targeted controls. And examples of these kinds of studies we've undertaken locally are shown on the slide. In terms of the methods that we have available, there are many methods, and it's important to understand them to kind of decide which ones are most useful to the end user. Usually they include analysis of genetic material, DNA or RNA, by molecular techniques to determine which human or animal source contributed bacteria or viruses to in the sample. Some methods really depend on the identification of physical and chemical characteristics of the bacteria that are exposed to different host species. Other methods really identify species or strains that are unique to a specific contaminant source. We usually split or classify methods into library dependent and library independent, without going into too much technical detail in these for these two categories. I would just say that the library dependent ones are based on databases or libraries of genotypic or phenotypic bacterial fingerprints, which we know are associated with specific fecal sources. And these methods usually require more time and are more expensive. The library independent ones identify sources based on known host specific characteristics of the bacteria or viruses and don't really need a library. So when we develop a sanitary profile, we can then tailor the FST study to the research question or questions that we have in mind and how much discrimination we need. We can use these recommended methods to guide us in this process. And what's important to note is that the FST study should be informed by the outcomes of the sanitary profiling and analysis of all the monitoring data that we have available, and should really address specific objectives. Ideally, samples should also be taken from multiple sites and sampling events during the period of interest, and during different weather conditions as well. So a few additional points that I just wanted to make in relation to FST methods. So they should really be used to complement, not to replace other methods that we have to identify contamination sources, such as monitoring it with fecal indicated bacteria. They are most useful to confirm the presence of a particular source or to better understand the types and relative abundance of the different sources. But there are limitations associated with method accuracy and for quantifications particularly, especially if the study area is affected by multiple sources. And because of these, the use of FST for source loading estimation and also predictive model calibration needs a bit more development. But it's clearly FST is most effective in giving us a qualitative understanding of the sources that are contributing more bacteria or sources that are more abundant in the catchment and thus need prioritizing. So the monitoring strategy that we have used at Cawthron has been based on the following 3 steps. So firstly, we identified the area of influence and contamination hotspots through field surveys and monitoring. In catchments where we have plenty of data, this could be just a simple desk screening assessment of the data. We then proceed to stage 2, where we carry out a detailed study of microbial variability in rivers or streams, and we usually target monitoring to represent the worst case conditions of contamination, looking at rainfall dependent discharges in the hotspots that we've identified in the desk study. And we do this through intensive sampling and sometimes using modeling. In phase 3 or in stage 3, we use source tracking studies to monitor the area of influence to give us a much better understanding of what's really affecting locally. And the question at this stage could be simply, are we detecting greater effects from human or animal sources, and to clarify really these contributions locally. So we use this phased approach in a recent study that we conducted in the Nelson for the Nelson catchment, where we've looked at long-term rainfall, river flow, land use, and microbial data for subcatchment areas, representing a range of land uses, and these are shown on the map on the left. We correlated the E. coli concentrations that we found in water samples from a set of sites across the catchment, representing the different land uses to kind of find the hotspots of contamination. So then we extrapolated these results to the wider catchment to quantify average concentrations for base flow and high flow conditions, and that's what's represented on the bar charts on the right hand side. So we see that, for example, high flow average results are higher than base flow ones, but for most sites, really the differences were not significant, were less than 1 log. The pie charts at the bottom of the slide compare the discharge volumes and the E. coli loadings for the Maitai River, the main freshwater inputs to the coastal zone, and compare that with the effluent discharge from the Nelson wastewater treatment plant. And what those results are showing is that during base flow conditions, the greatest E. coli loading is coming from the wastewater treatment plant discharge, while in terms of discharge volumes, there's really no or not much difference between these 2 sources. This slide now shows the results of the FST study, where we've taken water samples from the hotspots of contamination that we've identified in the area, in the main urban area in the lower catchment. And we tested for a range of markers indicative of contamination from human, ruminant, dog, sheep, pig, duck, and gulls. Most samples that we took from the CB site were positive for duck, gull, and human markers. And the samples that we took from sites YS 1 to 5, shown on the left, were mostly positive for human and most other animal markers. And these results have been really interesting and have provided information to the local council in terms of planning, sewage infrastructure improvements in the catchment, and identifying sites that, for example, are more vulnerable to issues like inflow and infiltration into the sewer network, and also to advise farmers on best management practices. So to conclude, so I have told you that sanitary profiling is a useful and valuable risk assessment tool that can be used to collect baseline information about the causes of water quality deterioration in primary production. Councils and farmers can also use sanitary survey data to better understand the potential health risks from exposure to pathogens. And that information it can also be used to improve monitoring. I've also shown that FST can be used in conjunction with sanitary profiling to identify sources that are likely most dominant in the catchment and focus management actions through more targeted pollution reduction planning. So just finally acknowledge partnerships that Cawthron Institute has maintained and developed with a range of organizations in developing new testing methods over the years, and also acknowledge funding received through the Seafood Safety Platform, research platform. This is all I have prepared for today. Welcome any questions. Thank you very much. There.
Thank you so much. So I do have a question on the chat, but I encourage others to put their questions on as well. So this is from Ali. Is it possible to distinguish between ruminants, e.g. cattle, sheep, with FST?
At the moment, not as far as I'm aware. And that is really a challenge, which is probably implicit in your question. When it comes to farm management practices and what farmers can really do to sort of mitigate bacterial loadings to rivers. I guess we could combine, and that's why sort of the observations in the sanitary profiling are valuable, because, as I said, it's really a complement of the two tools, or of both tools, really not single use of one tool or one coming later on in the process. It's really a conjunction that becomes, a lot more informative than just an MST, a one-off MST study, which provides little context to what's happening in the catchment in terms of livestock densities and species.
Thank you. And then we have another question from Philomena Ing. I think there are several human markers available. How do you decide which one is the best to use?
Good question. Again, the information from that initial desk-based assessment should give me clues on what is really affecting water quality in the area. For example, in this Nelson case study, we knew that the issues were associated with inflow and infiltration into the sewers. So we had a pretty good idea about that, also based on previous investigations. And that's where, again, the sanitary profiling and the shoreline survey can provide valuable information. Yeah.
Great, thank you, Carlos. We have another question from Peter Cressy, who's so far scoring twice, asking questions for both speakers. So Peter asks, is there potential for the FST methods to be made quantitative, e.g. percent human, percent ruminant, etc.?
There is potential, and we're working on it. We're having internal discussions on how best to provide this information in a way that is useful to the end user. it is certainly possible. I'll just be cautious about using quantitative information to validate models, catchment models, for example, or predictive models for recreational waters, for example. But a simple sort of apportionment estimate is possible, and we're working on it, again, bringing in and combining the information from the sanitary profile with the MSD. It's certainly possible.
Great. Okay.
Thank you.
That's all the questions. So thank you very much, Carlos. That was a really good presentation, and
thank you.
glad we were